Phenolphthalein, a naturally dangerous and potentially life-threatening substance. However, contrary to one's expectations, it was used across the globe and even in our school on a day-to-day basis for basic titration experiments as a pH indicator. Therefore, today, Clarence, Ivan, and I will be giving an introduction to our chemistry coursework that delves into the research topic of natural indicators, on whether anthocyanins found in fruits and vegetables, or more specifically, with onions, can be used as a natural indicator for checking pH. Firstly, what exactly are the differences between a natural and synthetic pH indicators? Well, things like litmus paper, methyl orange, phenol red, pentamethoxy and phenolphthalein have high costs, causes chemical pollution, while natural pH indicators such as Andrea flowers, litmus, turmeric, anthocyanins, and vanilla, which is an olfactory indicator, are cheap, environmentally friendly, sustainable, and non-toxic. Next, to elaborate on the dangers of phenolphthalein, a research study in 1996 used mice and rats as test subjects and discovered that the substance has the potential to cause tumors and cancer. The National Toxicology Program, Department of Health and Human Services, found that it begot genetic damage in mammals in the form of genetic mutations. Additionally, it caused morphological transformations, chromosomal aberrations in Syrian hamsters, which effects would definitely be transferable to humans. And thus, to ensure the well-being of any individuals conducting basic titration experiments, more people have been researching and exploring other alternatives, such as anthocyanins, found in fruits and vegetables, or in red onions for our case, as a pH indicator. Anthocyanins actually have an empirical formula of C15H11O+, with a molecular mass of 207.24724 grams/mole. It is a type of flavonoid and is a compound with antioxidant effects.
Now I'll be elaborating more on the characteristics of anthocyanin. So the color of the anthocyanin organic molecules depends on their pH. This is why they are viable as indicators. In strongly acidic media, anthocyanins are orange to red. In weakly acidic or neutral solutions, they are reddish violet or violet. In alkaline solutions, they are blue. Basically, this is because the number of hydrogen ions interacting with the anthocyanin molecule will change its structure. Differently structured anthocyanins will reflect and absorb different wavelengths of light, thus changing the color with which we view them. As of 2003, 400 types of anthocyanins were reported. Later literature in 2006 puts that number at over 550. Anthocyanins are synthesized via the phenylpropanoid pathway and eventually stored in the vacuole of plant cells. We can find anthocyanins in all tissues of higher plants, including the roots, leaves, stems, fruits, and flowers. In these aforementioned parts, they will be found in the outer cell layers, like the epidermis and peripheral mesophyll. As we were conducting research, we found a study in 2018 by a professor at the Serambi Mekah University in Indonesia. He conducted an experiment to extract anthocyanins from Syzygium cumini fruit and use them as pH indicators. He found that the extract of Syzygium cumini fruit can be used widely as an indicator of acid-base titrations in daily laboratory experiments that is environmentally friendly, non-toxic, and cheap. Another research study that we found was a study conducted by two Nigerian scholars in 2015. This study found that compared to phenolphthalein, the anthocyanins in ethanolic skin extract of red onion bulbs are actually an excellent substitution for the aforementioned substance, as a pH indicator with a standard deviation of 0.01 cubic centimeters in titration. In addition, it is cost-effective, friendly to the environment, and easy to prepare. So, based on the literature that we have reviewed so far, it seems that anthocyanins are indeed viable as a pH indicator. Now, I'll be elaborating on our chosen vegetable, red onion. Red onions contain anthocyanin pigments in their dried coats and outer parts of the fleshy scale leaves of the bulbs. The anthocyanin pigments in red onions are cyanidin 3-glucoside, 3-melanyl-glucoside, cyanidin 3-laminary bioside, and 3-melanyl laminary bioside.
So now we'll be moving on to the three extraction methods. So firstly, the first extraction method is the solvent extraction method. And this method is by far the most conventional method of the three methods I'll be presenting, and this is the easiest one. So firstly, the onions or any fruit or vegetable are to be grinded into small pieces. After that, you submerge the small pieces into either ethanol or water, or even a mix of both. Next, Another solvent that can be used is actually acetone or methanol, or even a mix of both as well. The anthocyanins will then be dissolved into the solvents after some time. Lastly, filter off the solid pieces of the solution and collect the filtrate. The filtrate can now be used as an acid-base indicator. So now on to the second extraction method, aqueous solution extraction method. So firstly, the plant, fruit, or vegetable material, or in this case, the red onion, will be immersed in hot water under normal or high pressure. Next, the extract will then be absorbed by non-polar macropause resin. Lastly, alternatively, it could also be extracted by deoxidized hot water, followed by ultrafiltration and reverse osmosis, concentrating it to obtain a crude extract that can be used as an acid-base pH indicator. So on to the last extraction method, microbial fermentation method. So this method actually involves biofermentation technology to extract the anthocyanins of the fruit, vegetable, or plant. So firstly, the microbial enzymes are used to achieve degradation of the anthocyanin containing cell wall. Lastly, the anthocyanins will then be completely dissolved into the extract. The extract can then be used as a pH indicator. So after gathering all this information, we have created our research question, which is, can anthocyanins from red onions be used as a pH indicator for volumetric analysis as a substitute for phenolphthalein? Thank you for listening to our presentation, and I hope that you have learned a thing or two from this presentation.